This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 830, recorded on November 16th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 44 degrees Fahrenheit, 5 degrees Celsius. It was quite a bit colder this morning, and it's a nice sunny blue sky, a few puffy clouds.
We have 9 degrees Celsius, partly cloudy. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's same temperature. It's 44 Fahrenheit, which is showing 7 degrees Celsius on my conversion, and it's actually kind of cloudy here. It's supposed to cloud over a bit, but not rain.
So, Alan, I took your advice. Last year you said you just cut the grass and that gets rid of the leaves. So I did that, and I have a lot of trees. So now I have this mulch all over the lawn.
Yeah. Yeah,
is that good?
It's good. It'll mulch down. I mow every week until about Thanksgiving. I actually have to go out. We just got it was really windy, and we got a huge load of oak leaves. We have a very heavily wooded lot too, and I just keep mowing them.
A few hours after I finished on Saturday, it was covered again.
Yeah,
with
of course.
leaves.
So you go out and mow again.
At least the grass was cut. And also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 79 degrees, and I would call it mostly sunny. There's some nice puffy clouds. Nice time of year. It's all good.
You have to clean leaves up, Rich?
Yeah, kind of.
Not much, huh?
It's not a big deal. I mean, a lot of our space is sort of beds that in the fall can just let the leaves mulch. Okay.
Yeah.
So there's just very little lawn. So what I do is just blow the leaves off the lawn into the beds and let them mulch.
If I remember,
clean up a little bit in the spring.
It's pretty small, your yard.
Oh, yeah.
So,
yeah.
Kathy, you have to clean your leaves?
I don't. I maybe I should, but I don't. I clean them off the sidewalk, and then I also clean them out of my street gutter, and sometimes I go in front of the house next door all the way to the storm drain at the corner. Otherwise, it is a mess when it snows.
Yeah.
Leaves, it just seems so futile, right?
Yeah, I remember in Buffalo, we had, we piled them out on the curb.
Yeah.
And then a machine that Niles used to call the Leafosaurus used to come and suck them up. And there was always this sort of game that you had to play about beating the snow,
Yep.
Yeah, but the mow mulching, I've been doing it for over 10 years now.
It works,
The key is you've just got to keep going about every week. And for me, it ends up being right around Thanksgiving, I'm done with the mowing. And then I don't participate in the whole leaf blower economy, which in this heavily wooded neighborhood, my God, it's awful. It's like being in a giant hornet's nest for a month and a half.
Right. That's absolutely right. Yep. And both Saturday and Sunday too.
But but it's noise and it's pollution and it's a complete waste of money. Just mow them, please, everybody.
So most people in our neighborhood get people to do this for them, and then they pile it up in the street, which now narrows the 2-lane to a single lane. And then if it snows, you're screwed because then it gets covered up and it's never moved.
A lot of the crews that go through here will have they'll have like 6 guys with the damn leaf blowers, and then they'll have a huge truck fitted with this giant vacuum thing, probly like
crazy.
The Leafosaurus.
That's the Leafosaurus,
and that's running in the background in the midst of the whole wah of the leaf blowers going on.
The Leafosaurus, I love it. That's great.
And that'll go on for like an hour, and then it'll stop, and you think, oh, great, it stopped. And then it starts up again right next, the next house. So
I was talking to Lynn Enquist, who's out in Arizona, and I said, I have to go do the leaves. He said, out here, we just move gravel around.
Yeah.
throughout November, December, and January, all of your donations made to Parasites Without Borders are matched and donated back to Microbe TV. Parasites Without Borders is a 501c3, so your donations are federal tax deductible in the U.S. You can go to parasiteswithoutborders.com, click the donate button. But I also have great news. This week I got a letter from the Internal Revenue Service. And I looked at it and I go, oh, crap.
Which is usually not a source of great news.
And I usually don't open them right away, but this was pretty thin. So it was saying Microbe TV has been approved as a 501c3.
Great.
So now, as of this month, everyone who donates via PayPal and Patreon, your donations are tax deductible and also going forward as well.
So we won't have to leech off Parasites Without Borders in order to get the tax deduction.
No, not in the future. And this should be a way for us to raise money to make a proper entity out of this company. In other words, proper in the sense that hire people and help out here and so forth. So it's very exciting. All right. 2 papers for you today. The first, as we were just discussing before I hit the record button, many people have emailed about this. For example, Mike wrote, I've been helping people with questions they have about SARS-CoV-2 and the COVID pandemic since the pandemic began. There's this paper in Viruses, gives the name of it. This paper has quickly been seized on by anti-vaccine and mandates groups as the newest reason not to use mRNA vaccines. All they see is DNA damage in the title, and it spreads like wildfire. Is there a way for you to cover this paper in your discussion? Interest in using this paper to discredit mRNA vaccines has quickly risen over the past 2 weeks. Particularly interested in what they mean by overexpression of the spike protein, how overexpressed. Keep up the good work. I could not do what I do without TWiV. So the paper is published in Viruses. Not a journal that we discuss all that much, but we do from time to time. It's an open access journal. The paper is called SARS-CoV-2 Spike Impairs DNA Damage Repair and Inhibits Vdj Recombination in Vitro.
Those last 2 words are really important. Sorry, go to the answers.
Hui Zhang and Yafang Mei from Stockholm University and Umeå University, which I was just thinking this morning, I'd love to go back to Sweden and, go to Umeå. I haven't been there. A lot of good virology there. Mm-hmm. And have dinner with Erling Norby again. Mm-hmm. Talk about Nobel Prize deliberations. That's always fun.
When you go, make sure you go to the library that has a really interesting clock. I can't tell you what library it is.
Okay.
We say that and they'll know exactly what it is.
All right, good.
I'll see if I can find you pictures and movies to send after the show.
So last time I went, you recommended I go see the Vasa, right?
That's in Stockholm.
Right. Oh, you're talking about Umeå. Okay.
Right.
Yeah, and what was I going to say?
The Vasa?
Yeah, the Vasa is the ship that they pulled out of the muck, and that was very cool also.
Mm-hmm.
I saw the Vasa in 1963.
Wow.
Ooh.
Pretty much right after they pulled
launched?
it up.
Yeah.
Oh.
Just barely after they pulled it up. I would love to see it again. It's probably different.
Yeah. I have a big poster of it in my garage.
Cool.
Well, in lieu of traveling to Sweden, we have a paper from 2 Swedish universities. And I thought that Rich and Kathy would enjoy it because it has to do with DNA and DNA damage repair, which is a feature of DNA virus infections. So what they have done here, I don't know how they got around to do this, frankly. I mean, they give a logic in the introduction, which is that, people with severe COVID have lower numbers of T cells and helper Ts and suppressor T cells, and there's delays in antibody production and so forth. I'm not sure that's what led to these. I just don't know, right? Nevertheless, what they wanted to look at is DNA repair pathway. So DNA often breaks in the cell, whether it's viral or cellular, and there are repair pathways to fix it because broken DNA, not a good thing to have. So these are called double-strand DNA break repair pathways. There is non-homologous end joining, which, as the word says, the two ends can just be jammed together without any homology. And then there's also homologous DNA repair. And then there is, and this is involved in making antibody genes, as we talked about a couple of weeks ago, the Vdj recombination. And that involves non-homologous end joining, right? So they wanted to know if this process is altered at all. But they didn't look at infected cells. They produced the spike protein of SARS-CoV-2 in infected cells.
They produced a few proteins, right?
That's right. They produced a few. Yeah, they produced a few. They produced some non-structural proteins, right? But I don't know what governed the choice. I mean, they do say that some of them are in the nucleus. People have shown before, and maybe that's part of it, right? NSP 1, 5, 9, 13, 14, and 16.
I was hoping somebody else would have some insight on why these particular experiments, because it struck me as kind of a weird thing to do.
I don't even know the function of having these proteins in the nucleus, right? You could imagine that they're doing something that we don't know about, but I don't know what that might. That would be more interesting than what that turned out here. But there's also a lot of spike. If you look at the immunofluorescence pictures, there's quite a bit of spike in the nucleus. Now, does anyone know if that happens in virus-infected cells? That would be an important question, no? If spike protein is in the nucleus?
Yeah.
Yeah, I don't know.
Because the spike gets targeted to the ER, right? It's a membrane protein. And then it goes to the ER-Golgi intermediate compartment. And then the nucleocapsid buds into that. And now you have a virus particle formed in the cell, which then moves by vesicular transport.
They have a sentence in the beginning of their results here where they say that some viral proteins are, they say, although SARS-CoV-2 proteins are synthesized in the cytosol, some proteins are also detectable in the nucleus, including NSP 1, 5, 9, 13, 14, and 16. And they give a reference. And I have not looked at that. So I don't know whether that is measuring localization of proteins during infection, which would be the appropriate thing to do.
Yeah.
Right.
One of the things that Mike asks here is what overexpression means. And I think we ought to detail that, because I think understanding that is critical to understanding the paper. And what that means typically is that you have a recombinant DNA molecule that encodes the protein that you're interested in under the control of some sort of a transcriptional promoter. That is something that'll promote synthesis of messenger RNA in cells. Okay. And you transfect that into the cells so that the cells take up this recombinant DNA and take it into the nucleus and transcribe it and translate it into protein. Okay. So that's typically overexpression. So these are cultured cells, and you're basically forcing the DNA into them and making a boatload of protein in those cells.
I don't know what the promoter is. I couldn't find it here.
I just looked up this reference for the subcellular localization of SARS-CoV-2 proteins. That is a letter to Nature back in November 2020. It's a very, very short report, and that is cloning the genes into expression vectors and expressing them in HEP2 and CACO2 cells.
So it's not infection either. Yeah.
So that's not infection either. So we don't have anything that's saying that any of this is relevant to infection.
Yeah.
That's not to say that assays like this are not useful and can give an indication what actually happens. There are, by the way, all proteins are made in the cytoplasm. So a protein showing up in the nucleus indicates that it's being specifically transported there.
Right.
And typically proteins that wind up in the nucleus have a signal for transport into the nucleus, though things I think can get into the nucleus fortuitously as well. Okay. Very importantly, we're not talking about infection with virus, even in culture. And we're certainly not talking about animals or humans.
Right.
Yeah.
Okay?
I don't know how overproduced these proteins are, right? I mean, you can't tell by the fluorescence because there's no comparison with infected cells. And, you can always turn up the fluorescence on your computer. So, that doesn't mean anything.
Yeah, so they put them into Puck 57 plasmid and transfected them into the 293 cells.
And I can't find anything about PUC 57 either.
Right.
In terms of a promoter or anything.
So, as Rich says, you can make a lot of protein by doing this. We don't know if they did. There's no quantification at all. But there is protein there. And the interesting part is that the nucleoprotein or nucleocapsid protein does appear to be limited to the cytoplasm. It's totally not in the nucleus at all, as opposed to all the others are clearly, I would say, more in the nucleus than anywhere else, right?
Mm-hmm.
Doesn't it look like that?
Yeah.
So why the function of that and whether there's a nuclear localization. I mean, if there's an NLS in each of those proteins, then I would say they're probably meant to go there for some reason, right?
That's a nuclear localization signal for those for whom the acronym escaped you. I'm sure plenty of precedent for RNA viruses sending proteins to the nucleus to tinker with things to benefit the virus infection. So it's not unreasonable that might happen.
Not at all.
Okay. But it's unclear in this case how that might relate to the infection or what these proteins are actually doing.
Even poliovirus proteins end up in the nucleus.
In this particular assay, the spike protein, a significant quantity of it, does wind up in the nucleus.
Yeah.
And that is curious. I would not have expected that.
And this is full-length, unmodified spike.
That's right.
Right, because it's what's made.
Yeah.
From their vector.
And there's no reason to think it would be processed by proteases the way it's being made, because that usually happens either at the plasma membrane or in an endosome, and these are not likely to be going through that. So it's full spike, uncleaved spike.
It is important to understand that this is outside of the context of a virus infection. I'm thinking about this because during a virus infection, the membrane architecture in the cell, in particular in a coronavirus infection, and many other aspects of the cell biology are completely rearranged.
Yep.
Okay.
Yep.
And so this is totally outside that context. So I don't, there must be immunofluorescence pictures somewhere of spike localization during an infection, but I'll bet money that it looks different than this.
Would you bet a dinner at that cafe in Austin?
Oh yeah. Matter of fact, I'll see your chili cafe, whatever, I forget what it was called, and
it was a cool
raise
place.
You something fancier.
Okay. I agree. I think that even at this point, before we get further into it, this is out of the context of viral infection, and it may be different. Yeah, it's a good point. So they want to know if this affects double-strand break repair. So they have a system where they have a plasmid that has two fluorescent reporter proteins, a green one and a red one. And I don't remember which is on all the time, and the other one has to be repaired to be turned on, right?
So it looks like the green one has to be repaired.
Yeah, so
it's in figure 1.
Do they cut it before they put the DNA in cells? Is that how it works? I think so. They say they cut it with SCE1 endonuclease, and then they put it in cells, and the only way the green is going to work is if it gets repaired by the double-strand break repair pathway. And they see in the presence of spike, the efficiency of both kinds of repair, the homologous and non-homologous repair homologous repair is diminished. So if you look at the graphs, they do this with all the proteins. And, empty vector is like almost 100% efficiency. Nucleocapsid is pretty close because it's not even in the nucleus. And then the NSP1 impairs this repair to a certain extent. The spike has the most effect, but NSP1 does it, NSP14 a little bit less, right? So, and both the non-homologous and the homologous repair. So, and the effect is about so it's about 25% of the levels without any protein being made.
And once again, did we detail how this assay is being done?
Not very, but you can do it more if you'd like.
Just quickly, because this involves co-transfecting in another plasmid that is arranged so that it's a target for repair enzymes. And if that target is acted on, that plasmid is acted on by the cellular repair enzymes, it has the effect of repairing a damaged fluorescent protein.
Yeah. So
that the cells fluoresce, and that gives you an easy readout. So it's I would say a somewhat indirect assay, though you are looking at the activity of the cellular enzymes.
You're looking at the activity of the cellular enzymes on an introduced target.
Yeah.
Yeah. And you're looking at it over the period of time of this assay, I guess, right?
Right.
Yep. Yep.
So if it's 25% as active, do we think that if we left it longer, it might finish the job?
Could
be.
I don't know. And the target is not likely to be chromatinized, not associated with histones, unlike the host
genome
cell DNA in the nucleus. Yeah.
They also find that it's only full-length spike that has this effect. I guess they put they produce the individual S1 and S2 separately, right?
Mm-hmm.
And they don't work, only full-length, which is peculiar. It gives it a kind of specificity, right?
Yeah.
Mm-hmm.
And then they looked at the amount of double-strand breaks using an interesting assay called a Comet Assay. Has either Kathy or Rich ever done a Comet Assay?
I never have. I had to look it up.
I haven't done it, but I have run across it in my past. So it's a measure of, you can, I think.
Go ahead. It's okay.
Well, you put the information in the thing. It's a gel electrophoresis assay where you embed your cells and you lyse them in the agarose medium that you've put them on this slide with. And then you electrophorese them. And if there's been DNA damage, you get a comet tail. Instead of migrating as a spot, it'll have a tail like an astronomical comet.
So you're looking at single cells, right?
Yeah.
So the single cells are moving through the agar in the electric field, and then, if there's some damage, you get a kind of a comet.
Basically, the way I see it is that the damage frays the DNA.
Yeah.
So if you apply an electric field, the little pieces migrate away from the bulk of the nucleus in a sort of a comet. And if you can, so they make this tail. And if after the damage and before the electrophoresis, any repair happens, it reduces the size of that comet tail.
Mm-hmm. So what they do is they cause DNA damage by treating with gamma irradiation, doxorubicin, hydrogen peroxide, which are known to cause DNA damage. And then they ask if that gets repaired with or without the spike protein. And the amount of repair is reflected in the length of the comet tail, I think, right?
Mm-hmm.
Here, I got a description from some paper the intensity of the comet relative to the head reflects the number of DNA breaks. The likely basis for this is that loops containing a break lose their supercoiling and become free to extend toward the anode. I like frayed. That works for me. It works for me. So there's an effect of a spike on repair of this, and they show you the pictures, and they're beautiful comets, but they quantify it as well. And let's see, tail length. So they're measuring tail length, comet tail length, and yeah, when you put the full length spike in, but it's not that much of a difference. It goes from 50% to 75%, right? So, half of the DNA is repaired, and 75% or 25% is repaired. So it's not that much. Although who knows how much you need to do anything, right? We're not working where we can say, yeah, 1 PFU is enough to infect the cell. We have no idea what these numbers mean in terms of these processes, right? I guess it's kind of like correlative protection. We don't know if 10% of the DNA repair pathway existing is still enough for a cell to go on normally, right? So then there's another experiment where there are proteins that are recruited to areas where DNA is being fixed in the nucleus. There are markers. So, there's one damage marker, gamma H2A.X, right? Boy, there's a memorable name. It becomes localized near the area in the nucleus, and they find that this localization is inhibited by spike protein. Markedly inhibits two of these proteins that they look at, forming foci, which is indicative of DNA damage repair. So it looks like the inhibition of repair is partially mediated by impairing recruitment of the repair proteins to the site of DNA damage, perhaps. Now, so part of the justification for this is that the Vdj recombination requires non-homologous end joining, right? Because the pieces of the antibody genes are chopped up and put together elsewhere by non-homologous recombination. As they say, we don't even know if the virus infects lymphocyte precursors, right? But they do have an assay. They designed an in vitro assay for Vdj recombination using antibody genes, and it's mediated by the enzyme that does it, RAG, which is a nuclease. And they say it production of spike inhibits Vdj recombination in vitro. And again,
this almost seemed like they had this assay sitting around, so they decided to use it.
This is another color assay where GFP is not green unless it's modified by Vdj recombination. Yeah, it does kind of. I would like to know the impetus for doing this. Yeah. Now, so those are the experiments, right? So there's some spike is going in the nucleus, which is odd, but could be real, but you'd need to look at infected cells. Seems to be impairing DNA repair, both non-homologous and homologous.
When it goes in the nucleus at these quantities, yeah.
Yes. It seems to be affecting Vdj recombination. And so they're discussing
using these reporters in this cell line.
Yes.
Yeah, all caveats, that's right. So they have this idea that perhaps spike is impairing immune responses. And so Rich had a conversation with Brianne about that. What did you learn, Rich?
So I'm pleased with myself here.
Yes, you got it right.
Yes, right. Yes.
So I said I had this correspondence with Brianne this morning. I have a theoretical question. The authors posit that inhibition of Vdj recombination during infection or vaccination could blunt the immune response. Does this make sense? I thought that the great majority of relevant Vdj recombination took place before antigen exposure, and that suppression of recombination following exposure would be of little consequence. And she wrote back and said, "You passed, dude."
Yeah.
Okay? That's exactly right.
So the immune system generates this whole diverse repertoire of antibodies against stuff you've never seen and stuff you may never see. And that's this whole VDJ joining thing, and as discussed on this show in previous episodes and on Immune extensively. And so that generation of diversity happens way back and, before the pandemic. And so you have somewhere in you, everybody has, unless they've got some immune dysfunction, antibodies against spike and against coronaviruses yet to be found and other, chemicals yet to be invented. So that exists in there, and that's generated partly through this VDJ or largely through this VDJ recombination type event. But that's a precursor of all this. It's before any spike gets anywhere.
Yeah.
So the horse has already left the barn.
Yeah.
Brianne went on, said that she looked at this paper and had a couple of other thoughts. First, VDJ recombination only occurs in developing B cells in the bone marrow and developing T cells in the thymus. For this to actually happen, that is the phenomenon that they're talking about to actually happen in an infected or vaccinated person, spike would need to get to those locations. And I'm not aware of any evidence of that happening either in vaccination or infection. And I can't imagine even a contrived way that spike could get there during vaccination.
Yeah. Yeah. And
I was just going to remind people that this VDJ recombination, as Rich already said, I think reading Brianne's answer, happens also in T cells. So it's not just the antibody rearrangement, it's the T cell receptor rearrangement.
Right. And on top of all that, when you are infected and recover, or if you're vaccinated with these vaccines, you make antibodies and you make B cell memory, you make T cells and you make T cell memory. There's no indication of any impairment.
Right.
Right. So.
And the vaccines work.
Yeah.
Vaccines work. This is not the issue. There are other issues, perhaps, but not this.
Yeah. This seems like a real stretch to explain. The immune findings in severe COVID-19 don't seem related to this at all.
No, I don't think so. No. My understanding, at least some studies have suggested that in people who go on to develop severe COVID, they have an initially modest interferon response, and that may allow more extensive virus reproduction. But the adaptive response seems to be fine in most cases,
So this is one of the general cautions that Alan brought up at the top of this, where he emphasized the words in vitro in the title.
Yeah.
This reminds, so it's a general caution about an important to understand the limitations of experiments done in cell culture. This reminds me, as we were saying in the pre-show, of the Yanish lab paper that we did months ago that seemed to indicate that at least in cultured cells, coronavirus RNA could wind up being reverse transcribed and wind up being integrated in the nucleus, and that set off a firestorm. But there's no evidence that goes anywhere beyond a contrived assay in culture. Okay, in vitro assays are, and similarly, hydroxychloroquine, right? Ivermectin, lots of other things that look like they might have some sort of an effect in vitro don't pan out. And so it's not as if in vitro assays are useless. They can point you in the right direction, but they should be interpreted with caution.
And in vitro assays are a great starting point when you're just poking around to see what's going on. They're cheap, relatively speaking. They're easy, relatively speaking. They're quick, relatively speaking. So these cells, they double in 24 hours. You can grow up a gazillion of them overnight. You can do all kinds of things with you, and nobody pickets your lab. You can they've got tremendous advantages, and you can do lots of biochemistry that you can't do even in tissues, and certainly not in intact animals or in people. So these are the kinds of experiments that are well worth doing, but you have to be very limited in your interpretation of what that means in vivo. It's just, here's something we found in cells. Now let's take that and try to go into more complex systems and see what's going on there. And my big problem with this paper is not necessarily that they did these experiments. I don't really understand some of the logic behind them, but fine, you're in the lab, you're dinking around with cells, and that's well and good. My big problem with it is the interpretation they apply to some of that, and particularly top line stuff like in the end of the abstract saying their findings underscore the potential side effects of full-length spike-based vaccines. You can't really do that. Just no.
So the first part of the sentence even isn't right. I would try to get in to say this. Our findings reveal a potential molecular mechanism by which the spike protein might impede adaptive immunity. We've just gone through why that's not the case.
No.
Yep.
And so then the second half of the sentence, which is what the anti-vaxxers are jumping on, does not follow, and I'm not going to repeat it.
No.
So in my mind, this raises, what are the responsibilities of the authors and of the editors? Okay, because you know damn well this is going to light a fire under the anti-vaxxers. Okay, it's experiments, it's data. They're reporting, real findings. Okay, the interpretation is wacky. Whose responsibility is it to do what?
Well, I think the reviewers should have said this last sentence is not appropriate, and you haven't discussed what Brienne told you and what you figured out at all. That should have been in the discussion, that this is irrelevant because the virus can't affect something that already happens before the virus gets there, right? That should have been in the discussion. So whoever reviewed it was remiss at not picking that up.
Right. The editor is listed on the front page, academic editor of the article. But I can't remember whether it's this MPDI journal or another one that ran into an issue 4 to 6 months ago with something being published and the editorial board being up in arms about how it must not have been reviewed very well or something.
Yeah, so, I think that had to be retracted even, right?
I don't remember how that panned out.
Something, we talked about this last time, last week, I think, or 2 weeks ago about, you remember there's this opinion piece in a journal which says, it's the one that you commented on, Kathy, in the emails that we read not too long ago, that, the vaccines are causing all these deaths and it's wrong. And there was a similar paper published that was retracted. I don't know if it's the same one you're talking about. But I think also this points out, in normal times, this would never get anybody's attention. With any other virus, the public wouldn't pay any attention. But because of the attention due to the pandemic, this kind of stuff gets, a lot of eyeballs. And so people don't understand the caveats and the limitations. But I do think I agree that the authors should have toned this back, and they should have put a discussion of why, even though this is happening. I mean, the data are there, right? They're controlled. There's something happening, but the biological relevance is probably zero.
Yeah.
And they should have said in the discussion, this probably doesn't matter, but the fact that it's doing this is interesting, and why the proteins in the nucleus might be interesting to figure out.
Yes.
They didn't.
Yeah, and they could even talk about subsequent experiments that ought to be done. Somebody should look in infected cells and see where this protein goes, yada, yada, yada. But I would assign blame across the whole pipeline. The authors needed to make sure that their conclusions are supported by their data. That's basic. Barring that, I understand authors want to make the work as significant as possible, and sometimes everybody's going to inflate things a little bit. Hey, this could mean this. But then it's the reviewer's job to step in and say, you can't really say that. And it is certainly the publisher's, the final editor's job before this goes to press, to look at it and say, wait a second, we're about to print something that says this in the current environment. Do the data support this? What did the reviewers say? Maybe loop back around through the review process again and say, are we cool with this?
Yeah, agreed. So anyway, for those who've been writing, this probably doesn't mean anything with respect to vaccination or infections in people.
Yeah.
Would that be a
what
fair?
happens over time with this kind of stuff is you have to be patient and see if there's any follow-up. Okay. And sometimes there's no follow-up at all, which by itself is revealing. Right. And sometimes there's follow-up that either supports or refutes the observations, because this is it. Science is self-correcting. And my guess is that over time, this will fall by the wayside.
Yeah.
So it's a word to those of you who are thinking of not getting vaccinated because of this. Don't get vaccinated. This should not impact your decision.
This has nothing to do with that.
At all.
Right.
All right. For the other course, I thought we'd return to remdesivir because we had such a cool, what was the name of that episode? It was a good title.
I looked it up. It was 819.
Oh, yeah.
What's the title? I can't remember. How to jam a machine or something like that.
How to stall a copy machine.
How to stall a copy machine. So this is published in PLOS. Pathogens in vitro selection of remdesivir resistance suggests evolutionary predictability of SARS-CoV-2. Interesting title. From the University of Glasgow and the University of Tartu, which is in Estonia. And
so the author list is really interesting because the first author and the last author, among about 20 or so authors, the first and the last author contributed equally to this work.
It's funny, yeah.
And my former UGA colleague is one of the authors, Massimo Palmerini.
Right, and Massimo has been on TWiV, as well as Emma Thompson from Glasgow also has been on TWiV. I like this because it is a very nice study in cell culture, mechanistic study, genetic study, and brings up some interesting questions.
And this is the kind of thing to do in vitro. This is a really good example of what you can do with in vitro studies.
So this is all about remdesivir, which, as we talked about, causes the RNA polymerase to stall. It is a nucleoside analog. It gets incorporated into the RNA that's being made by the RNA polymerase. And after the RNA moves a couple of positions, after it's incorporated, the drug clashes with one of the amino acids in the active site, and that's the end of it. You're stuck. Your copy machine is stalled. Very interesting mechanism.
The drug has the remdesivir has a big old bulky side chain on it.
Right.
That literally clashes with a residue in the enzyme. Okay. And can't go any further. What a great trick.
And as we, I think, talked about on 819, as we're likely to talk about with this paper as well, that's a great way to plug up replication in this virus in particular, because this virus has a proofreading activity. So this actually jams up the replication, and it doesn't matter that you can proofread for errors because you just shut down the enzyme.
So the introduction has a nice history of remdesivir, which had been developed before the pandemic. It was developed for MERS and SARS and Ebola. And a summary of the clinical data, which I've heard Daniel Griffin review for a long time now. And there's one study, 3 randomized trials demonstrated that remdesivir treatment reduced recovery by 31% and demonstrated a non-significant trend toward lower mortality. A non-significant trend. That's interesting.
Reduced recovery time, yeah.
Recovery time. And then a larger trial by WHO reported no effect on patient survival. So that's the last one Daniel reviewed. He said, that's it. I'm not using remdesivir. But they say despite these findings, countries still use it. And that US, UK routinely use it for patients who need oxygen. And as long as you have to be in the virus phase where there's virus reproduction, because if you're past that, it's useless to give remdesivir. Anyway, the point of this paper is, can we get resistance to remdesivir? Because there are no reports of remdesivir resistance out there. And a lot of people have gotten it. Kind of interesting, isn't it?
Yeah.
Yeah, so you can use this drug. It's not, you're not going to develop resistance to it, but it just doesn't work very well either.
Yeah.
So,
Good news, bad news.
Yeah.
So they say it's important to determine whether we can get resistance, where the changes are, and then monitor these in the population in case they start to pop up. So that's the paper, and it's really nicely done. It's a nice piece of work. So if you wanted to get resistance variants to a drug, what would you do? Well, you could passage the virus in the presence of the drug.
Yeah.
You infect cells in culture with the drug. You take what replicates. You then infect another culture, and you do that. They did it 13 times in 2 concentrations of remdesivir. And, of course, they have controls at the same time where they just passage the virus without a drug. And they look for cytopathic effect. And then eventually they do plaque assays, which is lovely. Another reason I like this paper. How often do we get a plaque assay?
How to get a paper on TWiV. Do plaque assays.
SARS-CoV-2 paper. So they get some resistance, not complete, but they get modest resistance, I think they call it. So the control virus is 5 log 10 units lower in the presence of remdesivir, right? So what number is that?
100,000.
100,000
fold less virus. Yeah,
so that's reasonable, okay? And then one of these lineages had a 2 to 2.5 fold increase in the concentration needed to inhibit 50%. And then there's a second variant as well that has a change in the 50% EC50. So they have two lineages, as they call them. The second lineage, 3.5 to 3.7 fold increase in the EC50. It's a little bit more than the other one. So you get resistance. And I think they say in the discussion, these arose very early on, I think after 4 passages.
Yeah.
Pretty quick. So where are the changes? They sequence the genome of these passage viruses. We have 2 non-synonymous mutations in the RNA, which means they code for a different amino acid. And they're not present in the input virus. They're not present in the virus passaged without drug. I mean, they do all the right things. Yeah. This is wonderful.
And this was, am I correct, that these mutations, we have here, they sequenced 3 independent isolates. Mm-hmm. Okay? And if I'm not mistaken, they found the same changes in all 3, these one that we're talking about.
Correct.
That's correct. That to me is quite significant.
Yes.
Yeah.
Okay. It's not just happened once. It happened 3 times in the same way.
Yeah.
So the first change is glutamine to aspartate at amino acid 802, which is in the RNA polymerase. RNA-dependent RNA polymerase, also called NSP12. And they say that a glutamate at that position is highly conserved among all beta coronaviruses, including SARS-CoV, MERS-CoV, and other sarbecoviruses. And this change, this amino acid 802, is in the palm subdomain of the enzyme where the active site is. They say it's in proximity to amino acids that are predicted to interact with newly synthesized RNA.
So it's right in the spot you would need to change to get around remdesivir.
Right. So I flipped out when I saw this because, and I had to look up the structures of the amino acids because I can never remember them. So these are both acidic amino acids. The structures are very similar. The difference is that aspartic acid is missing one carbon link in the side chain. Okay, so it's shorter by one carbon. And I immediately thought about the paper that we did where you could relieve the block by shortening the interfering amino acid from a serine to an alanine, removing one carbon. And I thought, holy cow, is this the same thing? But no. This is, which residue is this?
815.
815.
I'm sorry, 802.
802.
Sorry.
And the serine residue that we talked about before is 861. So, I mean, there's a lot of folding and stuff. So this is probably in the neighborhood, right? And they have a model that shows that. But it's not exactly the same as what we saw before.
So essentially, the idea would be that this change would allow the chain to keep going. It wouldn't clash with the remdesivir in it. It would not clash with the enzyme, right?
Right.
So
that's the idea.
This is a kind of resistance where the drug is incorporated, but it doesn't do anything.
Right.
As opposed to a change which would prevent the drug from being incorporated in the first place.
Yeah.
Which could also happen. All right. And then, all right, so that's one change. And we propose that this changes the steric interactions between amino acid side chains. So they're thinking that alleviates the clash, as we said, allows elongation when the active form of remdesivir is incorporated. So this is a perfect paper for following up to the stalled copy machine, right?
Yeah. Yeah.
In fact, now, if you want to teach this stuff, you could tell this story.
Yeah.
The clash and how to overcome it. Something like that. Now, the second change is in a different protein. It's NSP6. It's an isoleucine to threonine. And they say this is a protein, also highly conserved, involved in restricting autophagosome expansion. So autophagosomes are made during autophagy, right? When a cell is stressed, it captures bits of the cytoplasm in autophagosomes, it digests it, and then throws it out, kind of like recycling itself, right, before it dies. And so apparently this protein is involved in restricting autophagosomes, right, because the virus needs to reproduce before the autophagosomes take care of everything. This one is not conserved among coronas. They say we predict this may alter the structure of the transmembrane and extracellular domains of the protein. All right. So that's weird, right? I don't know why that would confer. Well, we'll see in a moment what happens. Okay, so
back on the conservation, did we talk about the conservation of the first site?
Yes.
Okay, in SARS, MERS. Okay, I missed that.
Beta, Sarbecos, Okay, so when you do this kind of work, you have to take these changes and reintroduce them into the virus and show that the recovered virus has the expected phenotype, right? And so they introduced either the 802D or A change into it. They recover infectious virus. They also did that with the NSP6 change, either alone or in combination with the NSP12 changes. All of these viruses reproduce similarly to wild-type virus. They use human lung cells, KLU3 cells. Kinetics are similar. Peak virus titers are similar. Okay, so without drug, there's no difference in reproduction. But the RNA polymerase change does confer partial resistance, about the same as what you saw in the passage virus. And the NSP6 change on its own didn't do anything. to drug resistance.
And if anything, the combination of the two was less resistant than the polymerase mutation by itself. Is that correct?
That's correct, yeah. Now, remind me, I've forgotten already. This NSP6 change, was that found together with the RNA polymerase change?
I think so.
Mm-hmm.
So it was fixed for some reason, but it has nothing to do with drug resistance? Is that what we're thinking?
I've got an idea, but I'm not going to reveal it yet.
Okay. No problem. Okay, so the RNA polymerase change recapitulates the partial resistance to remdesivir. Okay, so now the question is, do these changes confer a fitness cost? That is, do they impair reproduction in some way to the virus at the one change in the RNA polymerase? They did some one-step growth curves and they didn't see any difference, but a more sensitive way of looking at that would be a competition assay. So you mix the viruses, you can use two different ratios, and then they did use two different ratios, and then you infect cells, you take the yield after 24 hours, and you do another infection. They did this 3 times, and then you can titrate the virus. So you have to distinguish them by, I guess, sequencing, right?
Mm-hmm. Yeah. I think they do bulk sequencing, and you can tell the ratios,
right,
of the alleles.
Essentially, the wild-type virus outcompetes viruses with the 802 change.
Yep.
So
I thought this was a brilliant experiment.
This was a wonderful experiment.
Very nice. Yeah, it's important to do.
Wild-type kicks the mutant's butt if the drug is not around.
Yeah.
So I'm guessing the NSP6 mutation is some sort of compensation for this fitness cost.
You think?
Yeah. That's my hypothesis. So it would be interesting to do, and I don't think they did this, do the same experiment with the double mutant.
Yes.
Yeah.
The competition experiment.
That's a good idea.
Yeah.
They didn't, though.
Because you could do something entirely different to the infection to somehow compensate for the loss of fitness.
Yeah.
Okay. Mess around with the autophagosomes, right?
So you're improving fitness somewhere else to compensate. It has nothing to do with the original defect, right? That's cool. I like that. Very good. And then what they did next, which is very cool also, they say, okay, do we ever see this change in the billions and billions of sequences that we have?
Maybe not billions, but we have a lot of sequences.
844,376 as of March. Gosh, there are a lot of sequencing. And there's a database that you can go to look for that. They found out of those 36 sequences with that E802 change.
Is there any virus circulating in humans that's been sequenced more than this one?
God, I doubt it.
Do you think HIV has been sequenced? I was
thinking HIV is the only candidate I could think of that might, but...
There's a lot of flu sequences out there, too.
Yeah, but the scale of this, I mean, we're almost up to 1 million sequences from patient samples. These are clinical sample sequences. So
it's remarkable, yeah.
Yeah.
So the dominant replacement was E802D. And then they did see 4 E802As. Interesting. So the competition was done with D. They didn't do A. So they did. So there's some, but that's really few, and there's no onward transmission of these.
Out of those 844,000 sequences, they found 36 that had an E802 mutation. So 0.004% of the population.
So in other words, and one of them actually was from a patient in Scotland who never saw the drug, was not treated with remdesivir. So they say this change can obviously occur without drug selection.
And they point out that like half of American patients are getting remdesivir, and India is mass treating people with it. So it's not that the drug hasn't been used enough to have caused this mutation to take place. It's just not happening.
I wonder if it's the fitness cost, right?
I was thinking exactly the same thing.
Because, even though it's not a lot, it's in a population size, it would be probably enough to prevent this from moving, which is good. That's a nice thing to have, although the drug is not so great to begin with. If it were a great drug and you didn't get resistance, that would be great, right?
Right.
But
we have better drugs coming along now, and I'd like to see this paper done with the new antivirals that are just coming out.
I'm sure we will, yeah.
As a general principle, this idea of the trade-off of resistance and fitness is an important concept in antiviral drugs, because a lot of the initial antiviral drugs, for example, for Hep C and Hep B that were developed, ultimately didn't do so well because viruses could become resistant at no fitness cost.
Yeah.
So they could accumulate. The best drugs are those for which, if you can get resistance at all, it comes at a huge fitness cost. So you don't get resistance accumulating in the population.
And that's also part of the logic of combination therapies, where you're hitting from multiple angles, and the virus may be able to adapt fairly easily to one of these drugs or the other, but in the presence of both of those adaptations simultaneously apply some fitness cost that's too high to bear.
All right, and the last part is actually maybe even cooler.
Yeah.
So they say, okay, let's look in these passage populations. We have all these viruses we've passed, right? Let's look at changes not linked to resistance to remdesivir.
What other variants have we produced in this experiment? Yeah.
And remember, they're using a single cell line and 13 passages. Keep that in mind. There are 41 distinct non-synonymous mutations, 22 in all the sequences, and most of those were in spike. And those have been found in the in circulating viruses in people big time, unlike the remdesivir changes. These have been found, and you'll recognize E484K, that is in a lot of the variants, N501Y, et cetera. Deletions of 6970. These are amino acids in the spike. All of these have been shown in these variants of concern, and they found them here just by passing virus in cells in culture.
Yeah, with no selective pressure. These are just the ones that passage through.
Yeah.
Importantly, no immune selective pressure.
Yes.
Okay.
Yeah, no antibodies. That's right. No T cells. That's right. Exactly.
So it seems like this is just something, if you leave the virus to its own devices, it fiddles around with spike.
This is a cell culture with no immune pressure, right?
Yeah.
And these, arose relatively quickly. In people, it took till, September 2020 before we saw the alpha variant. And we'd been sequencing a lot before that. Why did it take so long in people?
People are more complicated than cultured cells.
Maybe.
Because in the presence of immune pressure and all the other, and the need to transmit because transmission is another thing that's not an issue in cell culture. You've got this researcher very obligingly pipetting you into the next plate. So you've got all these other constraints in the virus in people, and it took a while for it to develop a variant that grew out the population.
And these are human cells in culture they're using. So the way I look at it, this virus came from an animal, probably a bat or something else, and then it's not quite right in human cells. It's fiddling, right? And even in cells in culture, you can see that fiddling, right? And these are, so there are 5 codon. There's a 5 codon overlap. The changes in 5 codons are the same in their experiment as opposed to in variants. And they say the probability of that is very low. So there must be some common thing about fitness in people and in cells in culture that is selecting, but we don't know what it is. So it's not antibodies, as Rich said.
Yeah.
It's not antibodies. So that's really amazing to me.
And it tells you that this cell culture model of passaging these in human cells can predict the landscape that the variants are going to explore, and it gives you some right answers. It says these variants could arise.
I wouldn't have predicted that. I would have said, no, culture is too simple, right?
Yeah. And I think it's really revealing. I really like that result. As you said, it's one of the coolest results in the paper. And, it's clear they anticipated that there could be some sort of funny business going on. I appreciated their passage history of their parent virus when it showed up. They were they talked about where they got the virus, and that they passaged it just 3 times, and that they're calling the third passage P1 for their purposes, and blah, blah, blah. They were very careful to describe what this virus had experienced before it went into their culture system, so that they could make a legitimate comparison between that and the end product.
Yeah.
So they conclude, our data suggests that the parallel emergence of changes in spike and variants of concern were not necessarily due to immune-based selection pressure. So, all right, so maybe even in people, it wasn't immune-based,
exactly.
Right? Because that's what I was thinking too, and many people are thinking, right, that it's immune, but it's not. So the question is, what is it? What is the selection? And apparently you can use a cell culture to figure that out, right?
Yeah, it's something in the cell that's favoring some of these variants.
So some of these changes affect receptor interaction. So you could look at that. You could tease out these individual changes and ask what they do in cell. So cell culture is a nice system to look at that, right? To say, what are these? As long as you get a big enough phenotype, right?
Yep. Well, and you can do competition assays too, like they did.
Yeah, you could. Yeah.
Now it occurs to me that this doesn't mean that there is no effect on the immune response.
Right.
Okay. But that it implies that immune pressure is not selecting for these. That doesn't mean that there can't be an effect of these mutations on the
of course.
Immune profile of the virus.
In fact, there is, right?
Yeah.
We know that sera reacts differently with viruses with some of these changes, right? That's right. So I would love to know what was the selection for this in cells in culture. That may give you some clue about what was going on in people, right?
Yeah.
So I have a technical. Kathy.
No, I was just going to say one of the things I found interesting was that they didn't find anything in the XON, the proofreading exonuclease.
Right.
And also that they didn't find any bias of transitions or transversions, which is purines to purines, purines to pyrimidines or whatever. It's highly biochemical, technical thing, but they didn't find bias there. And from what I recall, they found it across the genome of these various mutations.
So I have an esoteric nit to pick for you budding or even mature virologists out there, which is, this sort of passaging and drug serial passaging and getting resistance and stuff, this is, I made part of my living doing this. And, plaque assays, one-step growth curves, all the rest of it. Anytime I see something like this, okay, I want to know what's the multiplicity of infection going in and what's coming out the other end in terms of virus particles or PFU, whatever quantity, per cell. Okay? When you report to me that you stuck in 1,000 PFU, actually, you could look it up and see that they were using, reported in their plaque assays that they used 12 well dishes and seeded them with something like 2.5 times 10 to the 5th cells per well when they did their plaque assays. So I assume that a 12 well dish holds roughly a couple of 100,000 cells. So if you have a couple of thousand PFU, that's a multiplicity of 1 in 100. That's important for this kind of stuff.
Yeah.
And in terms of what comes out, I'm sorry, PFU per mil doesn't give me any information at all. I don't know. Okay? You can do the calculation. It's really simple, and tell me how many PFU per cell did you get out the other end. That's the way you ought to report your plaque assays. In your one-step growth experiments, that's how you ought to report the passaging and everything else. Okay? So
people are going back now and thinking about their reviews and saying, Condit must have been one of my reviewers.
Well, and the reason you care about that is because it tells you how many generations of infection you're getting. And in this context, that's going to matter a lot.
Alan, you did a similar experiment for your thesis, remember?
I did, yes.
What did you passage polio at low temperature?
So I was working on, I didn't expect it to grow at 25 degrees C, and then it did, and then I had to figure out why, and that became my focus of my thesis. You did
identify the change, right?
Yes, I did. Right. And so I did a bunch of these low MOI and high MOI type of experiments, and thought a lot about how many generations of virus I had growing in a particular length of time.
And I remember the real challenge was to get an incubator that would stay at 25 degrees.
Because room temperature is not room temperature in any lab, particularly not in the Hammer building where room temperature was, anything across a 50-degree scale. And I actually, initially looked it up in the Fisher catalog, and you can get an incubator that'll maintain 25 degrees C. It's thousands of dollars because it's got to have a heating and an air conditioning circuit in it.
Yeah. Yeah.
And I thought about it, and I thought, well, what if I just put an incubator in the cold room?
Exactly. Yep. Yeah.
And I did, and it was okay, but it varied too much. It would, when it would heat up, it would get a little too hot, and I really had to stay at 25. So I took an old fan from a computer and stuck it in the incubator and turned it into a convection incubator just by plugging in this little fan. And then I got an NIST traceable thermometer, and I put it in there, and I verified that it was 25 on the dot. And so I had my 25-degree incubator in the cold room with the hacked computer fan in it.
To his credit, Alan did not ask me to purchase the expensive thing.
I didn't even go into his office about that. I was like, let's get a 25... Okay, let's figure out a different solution.
That's good. I appreciate that.
Yeah.
It's ingenuity. Very good. They make a statement here, which is interesting. Functional studies related to change observed during in vitro evolution can help to identify candidate amino acid residues likely to drive future adaptation. They're saying we should do these. We should do more of this passage and see what we get, because we might pick up something we haven't seen in people yet, and maybe we should look for it, right?
Yeah.
It could be important. So, you should. But if you propose that in a grant application, they say, it's not just fishing expedition, right? But it's what you should be doing.
Yes.
Science is tough, yeah. You got the right things to do are sometimes criticized extensively. Anyway, that's very nice, folks. Good job. And now you know even more about remdesivir.
Yes. And this being a PLOS paper, of course, is open access.
And why science is so beautiful when you sort out all this stuff, right? I mean, it's great that this is a virus causing a pandemic, but even if it weren't, it would still be really fascinating for us.
Yeah, you could do this with some obscure virus and we would still be interested.
I want to correct something. I think I misspoke when I said something about these mutations happening kind of uniformly. That's not really the case. They were much more predominant in the spike. So there was selection pressure for the spike. Yeah, positive selection pressure across. Yeah.
Let's do some email. Kathy, can you take the first one?
John writes, Vincent et al. Your comment that your wife pays your advice no heed brought this to mind. It's from George Beadle's autobiography, An Uncommon Farmer. So this is around 1932 at Caltech. Beadle, his wife, and Barbara McClintock are in his wife's Model A Ford Roadster. So 2 future Nobel laureates, and she was driving. There's seating for just 2 in one of those, so Beadle was riding on the running board, which was illegal on account of blocking the driver's view. They were pulled over. McClintock says, "Oh, it's okay, officer. You see, he's her husband, and she can see right through him." If I recall correctly, he said the cop let them go. Otherwise, dry and chilly of late in Greater Braddock. The last 3 or so nights have been below 0 Celsius. Sunny today and warming ahead. Regards, John.
That's a great story.
I like that a lot.
Could see right through. That's just great.
Yeah.
I love it. And McClintock was not known for being loquacious, right?
No.
Very cool. Rich.
Mauro writes, I started dipping into the archives and felt like watching those movie prequels where you find out how the characters came to be. Episodes like Concerto in B, the ones with Michael Minna, Shane Crotty, and Tony Fauci, they were not only fascinating, but they also happened to be the start of some current TWiV expressions and traditions, like restarting Pics of the Week, Racaniello, just whatever field, and more. There's a living TWiV culture. It's also funny to hear your thoughts back then and contrasting them with what ultimately happened, like when Vincent had doubts about the Pfizer vaccine working because it coded only for the receptor binding domain. Though, in fact, just a side note, Pfizer was floating 2 candidates at the time. One was RBD only, one was full-length spike, and the one that made the final cut was full-length spike. Thanks for keeping me sane, although we don't have the control of me not listening to TWiV this past year. So we'll never know if it was really you. Okay, fine. You get it. Let's say that you kept me up to date and provided an excellent example of authentic science talk. I can assure you, though, that you've made me healthier during my commutes, dishwashing, floor mopping, lab working, and I dare say some incident toilet sitting. Ooh, TMI. In Argentina, a famous singer saluted his crowd with a now classic beloved saying, "Gracias totales, Mauro, just a plant molecular biologist. P.S. I laugh every time you say epitopes. I wish you ended episodes with it every now and then."
What does it mean, totalis?
I think it means all the thanks, right? Right.
You can find a YouTube of it, so.
Yeah.
Yeah, epitopes is very funny. I keep mixing the two up.
Yeah, it's all the thanks or total thanks.
Okay, we're doing an epitope project in the lab, and often I say, which episode is that? Alan.
Fernando writes, hello from Germany, TWiV. See, not only viruses can spread far and wide. I've been a short-term listener, and my podcast app does not allow for any type of interaction, but the last episode with Gregory Zuckerman made me come to email to say that it was really good, and thank you for it. Best regards, Fernando in Bonn, who says, but I am Brazilian.
Should take another one.
Sure. Suzanne writes, my dearest lovely virologists, you have been a cooling salve for my burned and battered blue soul living in my deep red state, West Virginia, where it's cloudy, damp, and hovering around the freezing mark. I've been fascinated with microbes since I was in second grade and read about Joseph Lister, John Snow, and Louis Pasteur, among others. Alas, I am the daughter of high school dropouts. They believed education is for rich sissies and a woman's place is in the kitchen. So now, while I am working in a menial job, I have you with me. I have downloaded every episode. I have listened to all of them many times. I've learned so much. They say to do one step better than the generation that precedes you. My daughter has a master's in public health, and my son is studying engineering. I try to remember the words I do not know. I look them up when I get home. I wish you had a TWiV for dummies. I love when Brianna and Rich give the definitions as the podcast unfolds. Keep up the wonderful work. Much love and socially distanced hugs. Your devoted listener. Well, thank you very much, Suzanne.
Yeah, that's very nice.
That's very
lovely.
Nice.
Very lovely. And
that's exactly what we want to happen.
Yeah. And congratulations on the kids. Sounds like they're doing well. Mm-hmm.
I thought years ago about a TWiV for dummies, although not called that because it's copyrighted. But yeah, it would be interesting, right, to try and keep it really.
And we do
try
to. Rich and Brianne do the best job at this, definitely. Yep. But we do try to back up.
Yeah, like Richie goes, "What did I say today?" Some acronym.
NLS.
NLS. Yeah.
Yes.
Joe writes, I'm one of those who found you during the pandemic and have enjoyed learning from you and sharing what I learned from others, with others. I went back to the early days and am working my way back to the present, but also listening to the new ones as they come out. Bird flu episodes made me understand the lack of crows here in Cleveland Heights. I'm happy to report seeing them again, though still far fewer than 20 years ago. Dixon might say that was West Nile.
I would say that was West Nile.
Yeah.
West Nile.
I don't recall hearing specifically about this study on the show. I was having a discussion with a friend who is more concerned than I am. My position was that my current full Pfizer vax status is all I need, and when I'm with known vaxxed folks, I'm pretty darn safe. They are still worried about any infection and got boosted, but are still very cautious, concerned, and maybe think I'm being foolhardy. They stated that it is clear that vax effectiveness is waning. I asked what evidence gave them this idea. They shared this Times article, The Science of Vaccine Waning Immunity, which links to this study, which is effectiveness of the Pfizer mRNA vaccine up to 6 months in an integrated health system in the USA. What do you think about its claims? It does seem to say that we are still good against severe illness and hospitalization, but how are we against lower level infection? How are we for risk of catching COVID and passing it to others? So before I address it, it's funny that it's interesting that some people are going back and listening to older.
Yeah.
Right? Gosh, there are a lot of them.
Yeah.
It's harder and harder to do. Yeah. I did it when I started at around 92. And I only had to go and do the first 90 and then what was current.
Yeah, now it's
I can't imagine doing it now.
Apparently some people have. Isn't that great?
Yeah.
So this is a 6-month study where they have shown that, yes, effective. Now remember, these are observational studies now. They're not case controlled any longer, and there can always be confounding factors. But for fully vaccinated individuals, effectiveness against all COVID is now 73%.
All infection.
And infection.
Yeah.
And COVID hospital admissions, 90%. So effectiveness against infections went from 88% during the first month after full vaccination to 47% after 5 months. So that was, the other number was an average, I suppose. So, but what they say is that our results provide support for high effectiveness of the vaccine against hospital admissions up until around 6 months, even in the face of widespread Delta dissemination. Reduction in vaccine effectiveness against infections over time is probably due to waning immunity with time rather than the Delta variant escaping vaccine protection. And Brianne and I had a conversation about this on Friday. The term they use in immunology is contraction of the immune response, right? And she said, that's what we use because that's what it is. It's a natural contraction. Waning suggests that there's something wrong.
Yeah.
I wish people wouldn't use it, but it's stuck, right? It's stuck in there.
Yeah.
So I guess I would say that.
Yeah.
So the severe disease, hospitalization and death, that protection still remains high. Whether you can clearly be infected even if you're vaccinated. But the question is, how long can you transmit, if at all? Because apparently the RNA loads decline very rapidly, as we saw in the Singapore study. And now we're seeing evidence that you're not really getting infected, you're getting exposed. And there's a bit of virus in the nasopharynx, and it gives you a positive test. So I think the assumption that there is widespread transmission after vaccination is not correct. I think that eventually will shown to be much greatly reduced in vaccinated people.
And I think even if it is correct, it is irrelevant. These are COVID-19 vaccines. They are not SARS-CoV-2 vaccines. This whole thing is just expectation inflation. right? So these were developed and tested to prevent severe disease, and they are ridiculously effective at doing that. If you're fully vaccinated, your risk of ending up in the hospital or dead is more than an order of magnitude lower than it was if you weren't vaccinated. I mean, just you're great. The finding that people who'd been fully vaccinated didn't get infected nearly as often was pure bonus, right? That's gravy. All right, wonderful. This thing can actually slow down spread. That's lovely. That'll keep our emergency rooms and ICUs from getting overloaded while we finish vaccinating everybody. But then, okay, the immune contraction is you can't have all the antibodies at super high concentrations against everything all the time. So if you're immune from measles, you have, you got a lot of antibodies after you got the shot or after you got measles, and you got to ramp that down at some point because you just can't maintain that level. And so that's the immune contraction. And my thinking is at that point, yeah, you can get exposed and the virus can persist long enough to be detected and you'll pop positive on your test. And as Vincent said, it's not even clear that these are transmissible infections. But you're still out of the hospital. You're still protected, and that's what matters. And that's good at least 6 months, and I suspect it's going to be good a lot longer.
Could we do one more round, Kathy? Mark?
Okay. Hi, guys. Love the show. Very high quality and interesting topics and reviews. I have a quick question concern about Merck's new antiviral, Molna Piravir. I understand it is a viral mutagen working by causing wobble in copying viral template RNA, and with enough errors present, the resulting nucleic acid becomes non-functional. I also understand that the dosing recommended for this effect will be multiple times a day for multiple days, so 4 times a day for multiple days. As a retired pediatrician, I'm only too aware of the difficulties of getting families to take their medications as prescribed. Very often, 3 times daily meds turn into twice daily, and twice daily can be once a day or skipped now and then. Patients are only human, and outpatient dosing on a demanding schedule can often be difficult to adhere to. I'm wondering if anyone has studied the viral effects of administering low-dose molnupiravir. I assume the published TWiV regimen, so the dosing regimen, was selected for good antiviral effects, but I'm concerned that the real-life, i.e. lower-dose administration, will be possibly mutagenic and of low enough dosing to allow survival of possibly more robust viral variants. If the drug is used widely, even a relatively rare variant effect would then become more likely. Again, great podcast. I appreciate the time and effort you guys bring to the table.
The published QID 4 times a day regimen is.
Yeah, sorry.
I think it's a good point. I don't know if we can predict what's going to happen, right?
I don't. If there are data relating to this, I don't. I'm not aware of it.
No.
It is true that serial passage in low dose of an inhibitor can encourage resistance.
See today's episode.
At least resistance. With respect, so that's it's not a crazy idea.
No,
but and I also have trouble believing that the folks that reviewed and approved this drug didn't have this conversation. Okay, but I'm not aware of it, and I'm not aware of the data.
4 days is not that much, right?
No.
I mean, I can see if you have to take it for 6 months, then maybe people start to change. But I don't know. If this is somebody who's not vaccinated, they're probably happy to get an antiviral. I don't know. It's a good question.
There's a lot of crazy out there. And, Mark, being a pediatrician, I'm sure knows what he's talking about
of course.
In terms of compliance.
And this is a conversation about any kind of antimicrobial. This has been a problem for antibiotics for as long as we've had antibiotics, please remember to take the entire bottle. And yeah, this is a real concern. Do the patients actually take it on the schedule? And 4 times a day is a lot. And as we just talked about today, and as Rich pointed out, low dose over a period of time is a great way to select for resistance. And I could, based on today's paper, I could name a team of researchers who could probably look into this for you.
But as we saw today, you don't get remdesivir resistance in people. Maybe we won't with mole. Who knows? We'll see.
Or maybe we will. Or that's the experiment we're about to do.
I mean, I think someone has most likely done the in vitro molnupiravir resistance experiment, right? I'd love to hear it.
Yeah.
Rich, can you take the next one?
Bianca writes, "Hello, TWiV team. I'm writing from Townsville, Australia, where at 7 p.m. it is 27.1 C, 72% humidity. I am a lash technician, currently studying my Bachelor of Social Work. My job allows me to wear my headphones most of the day, and I have been thoroughly enjoying listening to the point that I considered changing my degree. Then I realized I probably do not have the brains for it. I got to stop there. Yes, you do. This is what can I say? This is not rocket science.
This is.
I don't know. If you don't have the brains for it, then I don't have the brains for it either. I've just been doing it for 50 years. Okay. It's not a big deal. Anyway, I found your amazing podcast last year around March when I needed the extra information. Having ADHD, I need to know every little bit of information possible about these things. And I can honestly say that your team has kept me well informed. I'm thinking of a person, an ornithologist, who described herself as, it's not ADHD. She, attention. I'll come back to it. I'll remember it. It's a joke. I may get lost every now and then. I also have to pause the podcast to Google a word, but in general, you're all fantastic at explaining terms for us non-scientific listeners. I have truly struggled through this pandemic in attempting to teach people how to research and make an informed and educated decision. And that's not research on Facebook and TikTok. I always refer them to your podcast, but what I'm finding is that they do not want facts. They want something to fit their idea of government control, medical freedom, and pro-choice, new version of anti-vax. We have been really lucky in Australia. We've had just over 190,000 cases and 1,882 deaths on the sunny state. My sunny state of Queensland has been most fortunate, and so far we have had 3 confirmed cases in Townsville with a population of around 185,000. That's amazing. And this is where the problem lies. Our vaccination rates are pathetic. I feel the fact that we haven't had to face the pandemic, bar a 6-week Australia-wide lockdown and a few weeks wearing masks, our lives have not really changed. But they will be on December 17th when rules come in for the vaccinated and unvaccinated. So my questions for the team are, we have a South Australian professor who has developed a protein-based vaccine, COVAX-19, and has been somewhat bragging out the mRNA vaccines. Are you able to explain the difference between the two? I feel he's doing it as he has developed a vaccine that isn't mRNA, but he has also mentioned that the technology is too new to know the long-term effects. I, along with my husband and sons, 20, 17, 15, and 13, are all fully vaccinated with Pfizer and blessed to not have had any side effects, bar sore arms. And in saying that, most people I know have not had any serious side effects. So my next question is, are the serious side effects, including death from vaccine, really underreported? Are they exaggerated? From what I have found, they seem to be within acceptable percentage alongside other vaccines. I have pretty much lost faith in humanity, and I am embarrassed to be part of such a self-entitled society with little or no respect for others. The unvaccinated minority seems to be the loudest, and I am constantly having to defend and justify myself for making a well-informed, educated choice in vaccinating myself and my family. Sorry for the longish email, and thank you for keeping me sane, informed, and inducing love for virology. Kindest regards, Bianca. Okay, so so protein vaccine, you have made the protein in a test tube. So it's there before you inject it, okay? And you inject the protein already made. An mRNA vaccine, you're injecting the instructions for making the protein, and the protein is made in you, okay? There are certain technical advantages in doing it that way. It's easier to get a vaccine quick because the technology is just easier to tinker once it's established. There's another advantage in that when the protein is made in you, you, without even an adjuvant, get a robust humoral, that is antibody, and a cellular response because of the fact the protein's made in your cells that helps stimulate the cellular response. Whereas with a protein vaccine, it may take technically a little longer to get around to actually making it in an emergency situation, as we've already seen. And you're going to need an adjuvant. Well, with some vaccines, you're going to need an adjuvant, okay, to help stimulate the cellular response. Not that that's a bad thing. There have been a lot of advances in adjuvants. Beyond that, sure, you can say it's an older technology, but there's no evidence that with any vaccine. I mean, there's this notion of long-term effects just has no basis in reality. All of the serious side effects that we've seen with vaccines show up within about the first 6 weeks of vaccination, which is why the trials were timed the way they were, so that people could be confident that any serious adverse effects that were going to show up had plenty of time to do so. So the likelihood that there's going to be long-term effects, this is just kind of a made-up thing. I understand the fear, okay? But there's no evidence to support the notion that it's going to be that way. So that's a pretty actually lame justification for comparing the protein and the mRNA vaccine. There's going to be advantages and disadvantages to either one. And up until now, when vaccines are becoming more available, what we've said is that the vaccine to get is the vaccine to choose is the one you can get. Okay.
Yes.
Because what you don't want is COVID. The idea of waiting until a vaccine is available when there's already one available is silly, because the worst thing that can happen is that you get COVID. So that's all I can say about the comparison of a protein and an mRNA vaccine.
I would add that in this particular case, the South Australian professor who has developed this vaccine, a counter criticism would be, first of all, that he has a rather obvious horse in this race, in bagging out mRNA vaccines. I guess that's Australian slang for putting them down. But also that presumably his vaccine is newer and hasn't been put in as many arms, whereas the mRNA vaccines have now been given to hundreds of millions of people around the world, and we're coming up pretty soon on a year of experience with these mRNA vaccines. We know exactly what their safety profile is. No question.
A year beyond phase 3.
Yeah. Yeah, we're well into phase 4 here, which is post-market. And there's no question about the safety profile on these things.
Which is a nice segue into, sorry, Kathy.
No, go ahead. Segue.
Nice segue into another part of her question is that are the side effects underreported? And the answer to that is definitely no. And probably where that comes from is I'm sure there's anti-vax trash talk that says, thousands of people have died from the vaccine. All you have to do is look at the VAERS, the Vaccine Adverse Effects Reporting System, and you can see that thousands of people have died after taking the vaccine, and that is a blatant misuse of that particular database, as we've talked about before. The fact is that when you take everything into account, as she suggests here in her email, what people might want to pin down as side effects, in fact, are not at any higher incidence in vaccinated individuals than in the general population. And the people who track this stuff are obsessive about reporting side effects and making sure that they see anything untoward going on.
Yeah.
The couple of cases in points are the thrombosis and the myocarditis, which were actually, in the long run, very rare. And in the case of the myocarditis, quite mild, okay? But they went so far as to pause vaccine distribution or trials when they saw these things come up, to be extra careful, which is, in my mind, proof that they're doing the right thing. And the last thing I would say is you don't have to defend yourself against people. I got vaccinated. Fine. That's it. If you want to know why I got vaccinated, you can ask me. Okay. And I'd be happy to explain my logic. But, you don't have to defend yourself. Though I can imagine that, depending on what kind of crowd you're exposed to, you can get people dumping on you. But, just ignore them. They're bullies. Just ignore them.
This Australian professor's vaccine it's also can be referred to as Spicogen. Phase 1 was in Australia, and then phase 2 was 400-person trial in the Islamic Republic of Iran. And phase 3 is about 17,000 people also in Iran. And that's the only country that it's currently approved in. So sometimes with the protein vaccines, Rich may have said this, they may be easier to make, transport, and so forth. And they have different pluses and minuses. But this one currently is not one of the 8 vaccines approved by use by the WHO. Maybe it will be someday, but not yet.
I'm trying to figure out where the protein is made. Does anyone know what kind of cells?
I don't know.
I didn't click on the clinical trial, so I don't know.
It says Oxford Expression Technologies. That's the name of the company. Anyway, it's adjuvanted for sure. Neat. It's been approved, authorized in Iran.
Yeah. In fact, the first, when you search them up, the first web page you get is in Arabic.
Yep. All right, one more. Alan, can you take that last one?
Sure. John writes, Vincent, Brianne, thoroughly inhospitable here in Greater Braddock. Clammy and overcast, less than 4°C all day, including light precipitation, some of which was in solid form. All I managed was trimming a few trees. But 829 made up for that. Most of my questions from yesterday answered regarding Paxlovid, very cool, and molecular warheads. I like that. I will be passing this along to my usual suspects and other friends, including one in the chemistry department, who has seen a decline in the performance of his students since the epidemic, to his despair. Are you seeing this at Columbia? It makes perfect sense that the protease would be highly conserved within coronaviruses. It has 11 sites that it needs to cleave with great fidelity. Any mutation in the protease is likely to be detrimental to cleavage at least one of those sites, which would wreck the whole process. Instead, if one site is getting cleaved suboptimally, evolution is going to favor adjustment of that site to better suit the protease. You also touched on UDP glucuronylyl transferase. It makes sense to me that would be found in olfactory mucosa. UDP glucuronylation, I knew I was going to screw that up, is a common mechanism for getting rid of poorly soluble compounds, and a lot of odorants, I think, fall into this category. The substrate for that enzyme, UDP glucuronic acid, is produced from UDP glucose by an unusual 4-electron transferring NAD-linked dehydrogenase, UDP glucose dehydrogenase. My old colleague David Feingold worked on that enzyme for many years, and nearly 30 years ago, my lab determined its amino acid sequence. Dr. Feingold was multilingual, and so this is just a preface to a great story about that, one that deserves a separate email to keep from diluting this one. Finally, the oak stake in the heart of that toxicological reports opinion thing is the great study that Daniel covered in 823 that followed 11 million people, 6.5 million vaccinated, 4.5 million not, and showed that the unvaccinated are 3 times more likely to die of COVID unrelated causes versus vaccinated. Okay.
Kathy, that's the toxicology paper that you commented on that was an opinion.
Oh, right.
Remember?
Right.
Yeah.
Yes. Mm-hmm.
So this is about the paper Brienne and I did on Friday where they talk about the development of Paxlovid, which was very interesting. But they mentioned this UDP glucuronylyl transferase. People who get anosmia have SNPs in that gene encoding that. That's the most prominent polymorphism in those individuals. And that is in the olfactory mucosa. But why it predisposes them to anosmia, not clear. It's an interesting thing. All right, time for some Pics. Kathy, what do you have for us?
I picked an article from the New York Times from several days ago that I've shared with several people. It's based primarily on David Leonhardt, the writer, talking to Dr. Robert Wachter. And he is the chair of medicine at UCSF and basically was asking, the headline in the paper copy is, when do we get back to normal? Maybe now. And so Dr. Wachter has recently begun to think about when things are going to get back to normal, and he believes the answer is now. So, for example, it says he's been eating indoors in restaurants and playing poker unmasked with vaccinated friends. He's flown to visit relatives. He hosted a conference in downtown San Francisco with a few hundred masked and vaccinated attendees. And so, of course, as always, it depends on your particular site and prevalence. But for instance, in Seattle, the daily COVID hospitalization rate has been slightly above 1 in a million. By comparison, the flu hospitalization rate in a typical year is more than twice as high. And so for most vaccinated people in a place like Seattle and San Francisco, COVID already resembles just another illness. And so this article goes on to say that basically, if we don't have some kind of benchmarks for when are we going to do certain things, we will just sleepwalk into policy. That was a quote from Joseph Allen of Harvard. And so, for instance, saying when, maybe when 6-month to 4-year-olds can get vaccinated, or, when levels are at a certain point or something, then we can think about not masking and so forth. And so I just thought it was really, really well done. He says, for example, he'll continue to wear a mask on an airplane and at the grocery store because there's not a very high cost to that. But when it comes to speaking and communicating or other things, maybe some of those mitigations should be reconsidered, in particular, like not having school in person, things like that. So it just really brings up a lot of points. He interviews a couple other people as well.
Interesting. Yeah, we still have to wear masks at Columbia to teach.
Oh, yeah.
Which just makes me speak louder. It's actually okay.
Yeah, the state policy in Michigan is if you're giving a speech, then people are more than 12 feet away from you, you can be unmasked. But if you're teaching and the same distance were to hold true, that can't be the case. So, we give our seminar in the same room as the student seminar on Friday because the student seminar is a class. They have to wear masks. So.
I think what a lot of people don't appreciate is that we don't know the answers to a lot of these questions.
No.
We're still. I mean, people who've been listening to back episodes, okay, are amused by the evolution in our understanding of what's going on, and there's been a profound evolution in everybody's understanding of what's going on, and we're not done yet. Okay. I would kind of agree with this if I look at the local statistics and I think about vaccine rates and all that kind of stuff. I'm ready to say, okay, except for certain circumstances, I'm pretty much done with this. But the last time we did that, we got another spike. Okay, and it was huge, and we're still a long way from being fully vaccinated or fully immune. So. I'm not saying there's going to necessarily be another spike, but who knows? We don't know. That's the point. We don't know. And what frustrates me is that people's expectations are so high. They expect all the public health officials and everybody else to have the policy right every time and have all this kind of stuff figured out, and there'd be no surprises, and don't change your minds and all that. I'm sorry, that's not the way it works. We're making this up as we go along because we have to.
The end game of this, I'm kind of projecting January because that'll be the point where kids
for the U.S. You mean
for the U.S. That'll be the point where kids 5 and up and all adults will have had the opportunity to become fully vaccinated. At which point, I'm kind of the opinion that if you're not fully vaccinated by that point and you had every opportunity to be so, you're on your own. You're not my problem. It just, you had your chance to do this right and you chose not to. And if you're going to be like that, then you're taking your own risk. I think the insurance companies ought to be let off the hook for COVID-19 related expenses for unvaccinated people at that point.
That would get some vaccine.
I think that may get some vaccine uptake, but my feeling on this is I don't see why my premiums should be covering somebody else's willful stupidity at that level of expense. And I know there's not going to be any policy traction for that, but that's the kind of thing that I would actually support. And under, you could still say, yes, kids under 5 will not have been vaccinated at that point, but the statistics on that, I mean, they're real low in the risk category. And that's not to say that it doesn't matter that some young kids can get badly hurt by this. But a little thought experiment I've been trying to do lately is how many people die every year from the 4 common cold coronaviruses? Before you say none, ask yourself how we would know.
Yeah.
Now, every year, many people die of viral pneumonia, and nobody sequences those viruses. The 4 common cold coronas circulate. They cause viral pneumonia. Somebody's 95 years old in a nursing home and develops that and dies. I don't think the death certificate is even going to go into much detail about what happened. Right?
Yeah. So my guess is more like March, because I think by then we might have data for the 6-month to 4-year-old population, and we would be past perhaps a winter peak. And of holidays and all that kind of thing, and be coming back into the warmer season where we could do things outside in the U.S., in the northern part of the U.S., things like that. I don't know. We'll see.
Yeah, we will. Rich, what do you have for us?
So this is probably at least partially redundant of past picks. My pick is a New York Times regular feature, regular sort of subsection called the COVID-19 pandemic, which is a real-time comprehensive resource for COVID information. I think we've picked subdomains of this at various times, but I've come to, I was looking for sort of the top level link that could get you to everything else without too much fishing around. And so that's this one. It looks like just sort of news headlines and stuff, but in particular, if you pan down, my favorite part is maps and trackers. and because the map is on the front page of the digital New York Times every day about COVID incident. And I think what finally did it for me for picking this were two things. If you look under maps and trackers and goes down, it talks about other trackers, one of which is mask mandates. They've got a map of where there are mask mandates and where there aren't. And if you're feeling really frisky, you can go and compare that with the case incidents and the death incidents and that kind of stuff. I haven't done that myself. But there's a whole bunch of data here that's really very well done and very nicely displayed that I really like. And the other thing that got me, of course, vaccines, I have picked that before. Their vaccine tracker is excellent. But they've got one that I hadn't seen before, which is treatments. Okay. So these are antivirals and et cetera, where they tell you what's been FDA approved and also what's widely used, not necessarily
the same list.
Things that are promising evidence, tentative or mixed evidence, pseudoscience or fraud. Okay. And you can actually sub-select those. Because I was interested in, I got into this because I was interested in the status and a summary of this new dude, Paxlovid, the protease inhibitor, which really looks great. So that is Pfizer, retooled from an earlier SARS drug. Cut the risk of hospitalization and death by 89% when given within 3 days of the start of symptoms. I don't know what the dosage is on that, but that's amazing.
Yeah.
Okay. At any rate, that's an aside. I think this is a wonderful, if you got questions about COVID, go to the top page here. I'll bet you'll find some answers.
It's a great starting point.
Yeah.
Mm-hmm.
I think it's a multi-day dosing, just like we were talking about. Yeah. So you got to stick with it. Yeah, we talked about the development history of that drug on Friday. It's very interesting. The development started before the pandemic.
Yeah. It was
well before
SARS and MERS, I think.
Yeah, just like Molnupiravir.
Mm-hmm. As a matter of fact, sometime we ought to go back and do a whole protease inhibitor history.
Yeah, sure. That's a good idea. I'd love to. Yep. Alan, what do you have for us?
I have, okay, so this is kind of out of the blue, but I was shopping around for some rather obscure tea. I'm not normally much of a tea drinker. I have coffee, but in the process of, one of these internet search things, you're looking for one thing and you end up finding something totally different. And I saw this company that had American tea. I was like, do we grow tea in the U.S.? I guess, they grow wine in California. They're fine. But it turns out this is actual American tea. It's a native plant. And this may grow outside Rich's house now. I'm sure it grew near your place in Florida. The Yopan Holly, which is native to the whole southeastern U.S. It's very widespread, and it is a naturally caffeinated plant. It's the only naturally caffeinated plant in North America. It's related to one in South America that's used to produce yerba mate, which is a trendy tea thing. But this thing, it turns out, was not just recently discovered to be naturally caffeinated. It was a core part of the cultures of indigenous tribes throughout that region, was referred to as the black drink. I think some of these tribes would get together and drink massive quantities of it and get totally hyped up on the caffeine and then, go have parties or battles or whatever. But it was a big part of their culture. Which, of course, was lost when Europeans arrived and wiped those folks out. But then this company, Yopan Brothers, has set itself up to harvest and dry and rediscover this and distribute it. And I got some of it, and it's good stuff. It's got a little bit, a very small amount of caffeine. It's about like green tea. And they've got a few different flavors. There's one that's just a basic green tea that's dried. There's one that is that's fire roasted. That I think that may be the traditional Seminole way of doing it. That's got kind of a smoky taste to it. It's got the antioxidants that are found in other teas if you're into that, but it does not have any tannins, which is neat because you can brew it for several minutes and it has no bitterness to it whatsoever.
Which one did you get?
I got a sampler pack, and I have tried. I gave a couple of them away as gifts, but I've tried the green, what they call the Warriors tea, which is the fire roasted one, and the mint. Actually, my wife really likes the mint, and I think that's it. Oh, I think I had one of the chai, which is with some additional spices.
I'm going to get the chai. I really like chai, and I want to try. I need some tea here at the incubator.
Absolutely.
I'm going to get that.
We sustainably harvest the leaves for our teas locally from organically certified property in New Smyrna Beach, Florida. Who knew?
Yeah, Florida chai.
New Smyrna Beach. We used to sail down to there. They've got their municipal yacht harbor has the best showers on the Florida coast.
Yeah, and apparently this stuff is it just grows wild. It's practically a weed that just grows all over the Southeast, and people use it in landscaping, or it just grows in the forest.
Cool. And now, of course, I need to get a water heater because I don't have any hot water here. There's no stove, so get one of those plug-in things. Now you're costing me money.
No, just get a little like an induction plate or a kettle or something.
Yeah. I mean, I need. Well, then if I do that, then I need a vessel to heat it in, right?
A teapot. That's not that much money.
If I get one of those plug-in electric thingies that are ubiquitous in the UK, then that'd be fun. Cool.
Yeah.
All right. My pick is installment number 8, Seminal Papers in Molecular Biology. So this one we had mentioned before. Rich had talked about this. This is a PNAS paper from 1961. And if you look at it, it's ancient history. It's so different. The dependence of cell-free protein synthesis in E. coli upon naturally occurring or synthetic polyribonucleotides by Marshall Nierenberg and Heinrich Mattei. So this is the beginning of deciphering the genetic code. And they developed a E. coli in vitro translation system, which was not trivial. They show that you can get it to make protein. And then in the first of a series of classic experiments, they in this case, they programmed it with synthetic U, lots of U's, poly U, right? And they looked at the protein that was made, and they said, whoa, it's phenylalanine. So U must code for phenylalanine.
Phenylalanine.
And so they ended up going through all the different synthetic single and double. In fact, there's a nice review article in Trends in Biochemical Sciences. It's called Historical Review, Deciphering the Genetic Code, a Personal Account. This is Marshall Nierenberg. And they have a table in the paper where they show you, so U gave you Phe, C, poly C gave you proline, poly A gave you lysine, G nothing. And then they use
G precipitates.
Precipitates,
you
okay.
Can't. Poly G is really a crappy homopolymer.
Then they tried UC, UA, UG, CA, CG, AG, and then finally UAG and CAG. And of course, the synthesis was pioneered by H.G. Khorana, right? So that made this possible. And they ended up getting the Nobel Prize for this in 1968 to Khorana, Nierenberg, and Robert Holly as well. This is a really nice firsthand account by Nierenberg, who just died a couple of years ago, actually. I have to say, when Erling Norby visited Columbia in, I think, October 2019, right? And he gave a talk about, and the talk had something to do about the genetic code in it, right? So this lady shows up and she said, I was Nuremberg's last wife. Because he had several, and she was the wife he had when he died. And so she really wanted to hear about it. But she left after 10 minutes. I guess she wasn't so interested in it. But
she just wanted to say that.
Yeah, maybe. But anyway, this is so cool, this personal history. I would like to tell you about how the genetic code was deciphered. He was a postdoc at NIH after getting a PhD at Ann Arbor from the University of Michigan, which Kathy, of course, knows. All right. The next year, I started work with William Jacobi, and I isolated the pseudomonad that grew on gamma-butyrolactone, and I purified 3 enzymes involved in the catabolism of gamma-hydroxybutyric acid. I mean, this is just what people did, right? They purified enzymes and learned how to do biochemistry. And then he goes on to talk about how he got into this and the things that needed to be done in the history of the field. It's really good. It's really very nice.
This looks really great. This is very nice.
And, people giving talks and going to meetings and hearing things that led him to do this. After returning to Bethesda, Fritz Lippmann generously gave me a partially purified transfer enzyme, and we showed that phenylalanine is an intermediate in the synthesis of polyphenylalanine directed by poly. It's just so cool. And all these old pictures with people writing on the wall and stuff. So, and then I love figure 8. This is great. A going away party where everyone is dressed in 60s gear. The men have skinny ties, and the women have these pencil skirts. Oh, it's so funny. And the colors are faded. So very cool, very important, obviously. The paper is incredibly straightforward.
Unfortunately, it is paywalled.
I'm sorry about that. I thought I found it on PubMed, the free PubMed thingy.
The link for the historical paper.
The historical is paywall, but not the original.
Oh, the original, the original you could just view.
Anyway, so that's that. And then we have a couple of listener picks. Justin writes, GOP is a death cult. It blows my mind. There are still people who would rather get sick and die or get their family and friends ill than do something that's free. And he links to a CNN article, which Kathy has fixed the link to. Flu shot uptake is now partisan. It didn't used to be. Yeah. So they did some surveys, and uptake is politically falls along political lines. And then he had, at the end of his email, has a quote by Isaac Asimov. There's a cult of ignorance in the U.S., and there always has been. The strain of anti-intellectualism has been a constant thread winding its way through our political and cultural life, nurtured by the false notion that democracy means that, quote, my ignorance is just as good as your knowledge, end quote. That was from 1980 in a Newsweek article. So this is not new.
Yeah.
Nope.
It's funny, Lex Friedman, before he started our pod, he said, don't show off that you know things. People don't like that. I'm like showing it off. What's wrong with having 40 years of expertise in a subject? I mean, but he said, no, that's not how you can reach people. You have to be very humble. Oh, okay. I said, I never do it anyway, but I don't know what the problem with knowledge is, right? The death of expertise.
Yep.
And then Lisa writes, I enjoy and benefit from all you do. Thank you. I think your listeners might enjoy the podcast Criminal, and especially episode 176, The Red Flag. It's about an investigation into a series of hepatitis C infections, and it shines a light on how insufficient human resource policies and inaction can rip huge chunks out of even our most trusted and admired institutions. This sounds great. Very good. Thank you, Lisa. Lisa's from Virginia. All right, that's it for TWiV 830. You can find the show notes at microbe.tv/TWiV. You can send us a question or comment, TWiV at microbe.tv. We do love hearing from you. And remember, throughout November, December, January, If you donate to parasiteswithoutborders.com, they will match the donation and provide it back to microbe.tv. And of course, if you want to donate directly to Microbe TV, you can go to microbe.tv/contribute. Your donations are now federal U.S. tax deductible. Kathy Spindler's at the University of Michigan, Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
You're going to have a new governor there?
I hope so. Oops.
Oh, boy. Alan Dove is at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.